This is part of Module 3, Structure-Based Evidence. You should have already done a TigerFAM, a PFAM analysis for your bacteria, for your gene. And what that leaves for us to do in the last part is a PDB analysis. And PDB is a protein data bank. The instructions start on page 60 in the training manual. And the protein data bank is highly curated, which means that it is a really specific, well-tended database. For a protein to get into this database, it has to be homogenized, crystallized, and X-ray crystallographized. It has to have an X-ray crystallograph taken of it. So it's got to have been experimented on in a lab to establish its function a little bit more. It's not some random protein. So being highly curated means that if you find something here, it would lead you conclusively to believe that your protein has those details and it's an authoritative source that's saying it. On page 72 is where the procedures start for protein data bank. So I am already in my notebook for KSED 00010. I'm going to control click to open a new tab, and the first thing I need to do is click advanced. And I am going to do a blast FASTA option from the sequence features, and I'm going to get the FASTA stuff from my notebook. So I've got to go back to my notebook up to basic information. We saved this here. I'm going to make sure I get the amino acid sequence including the FASTA header. Copy. I did Control C, Control V to paste, and I'm not going to touch it and reorganize it or anything like that. It should read it properly just like that. And I'm going to do the e-value cutoff. As usual, we don't want 10. We want 0.001 as usual. And we're going to do a search or submit our query. This is running our sequence against all other sequences in this data bank. So when you get your results, you can see what this says for KSED 00010, but you have to do your own analysis for Clostridium. So you're going to look at the quality of the alignments between your query gene and your blast hits in PDB. And remember that e-value is going to be critical. The 0.001 or less means that it's not likely to chance if it's less than 0.001. And on page 73 of your training manual, it says, if the e-value meets the cutoff set by the instructor and a significant length of the protein is aligned, then it's a good hit. If two proteins are similar in sequence and have approximately the same length, it's highly probable that they fold very similarly. Therefore, structure corresponds to the PDB blast hit, most likely resembles how the query gene product folds. If you get one or more good matches, record the code, name, length, score, alignment length, and e-value in the lab notebook. And it starts to tell you that in the training manual on page 74. So let's look at the results that we just got. This is the top hit. And notice that it's sorted e-value best to worst. So the top hit is this 2Z4R entity 1 containing chain A, B, and C. And they've got the organism that it came from, and it's a replication initiation protein DNA A. This is a name that has been coming up with KSED 00010 for a long time. So we're just building evidence that our protein in Chytococcus sedentarius is probably analogous to this protein or homologous to it. But you can see all these details. They have experimented with this in a lab to establish these things. This gives some details. The e-value is definitely below 0.001. There are a couple gaps, but not many, and you can see that with X's. So back in the notebook, I'm going to go back down to the PDB section. The first thing it wants is a PDB code, and that is going to be this right here, this first section. So not the whole link. You can just take the code out of the database, save it. You can include information. More information is better than not, but here's where they separate out the name anyway. So I'm just clicking on this to expand it out. I'm going to copy the name. Save it. The alignment length, the 346, copy it, Some of this stuff you have to just look around carefully before you get overwhelmed at the amount of information that it's presenting to you. So just save it. And the e-value, we always get the e-value, which is also listed there. Just look carefully. Copy, paste, save. And the pairwise alignment, we are going to try to get this whole thing. But getting this into the notebook, I mean, this is not going to be very easy because you have to scroll. So there is an easier way built in that might be available for your protein. So if this isn't available, what you would do is you would start at the end of the alignment and do like I showed you in another one in a trick. You'd take a snapshot, maybe from 400 here to 505. You'd insert that into your notebook first. You'd do the snip tool and you would put that in. Then you'd scroll over and do a little bit of an overlap so that maybe it was 290 to 400. You'd insert that next. Because remember, you start at the end because when you upload a picture into GENI-ACT, the first picture will get bumped down as you add more pictures. But if you have this option, it's a little easier. Select a report, and you're going to pick the Blast FASTA PSI Blast results. I have to allow pop-ups, and here's something here. Now remember, we are looking at Z2Z4R. So I want to find the 2Z4R. This is 2Z4S, and here it is. So what I'm going to do, now this is snippable. Open the snipping tool, a new snip. I'm just going to try to get everything from this. I'm going to save it. I'm doing it to the desktop. You would do to wherever you're keeping these things. You might want to insert a picture on your poster, so you need to be able to get to these again. Go back to GENI-ACT. Pairwise alignment, and this time I am browsing for that picture and I'm uploading that alignment. Save it, and I'll double check it quick. Remember, you can drag this open. So one last thing you can do is if you click on the first hit, you can see if there's any literature about or further information about it, and in this one there actually is. And you can actually read about it. And you can see just a little abstract here that talks again about what this DNA-A does. And this is kind of a big deal for you because at this point you might be ready to say that, well, my gene is most likely doing this. Or it might be really strongly affiliated with this other gene that I found in this databank. So at this point, at the end of Module 3, compared to this structure-based sequence, or similarity module, and the sequence-based similarity module, module 2 that we did, you might be able already with your Clostridium gene to guess the name and function of your protein based on evidence. If you can't, especially if your protein is hypothetical and nobody's really quite sure on any of the things and you're really doing the first legwork with it ever, the other modules might help. But you should be getting pretty close. These literature citations, like what you see here, are excellent things to include on your poster. So as you go back through your notebook and you see, how do I know this? If you can use those sources, it makes your poster professional. This last thing lets you see the structure in 3D. I just picked the in-browser option, and it lets you mess around with it a little bit and see what this protein, how they think it actually folds up. You can see a bunch of helices here, the little curlicue spirals, and it just lets you play. Take a snapshot too if you want. You can include that in your notebook. But this is actually what this DNA-A folds up as. So lots of cool stuff as you start to put the picture together on what your protein actually might do.